Hello dear aspirants, welcome to the GDC Classes English, especially for specially designed Pharma Thunderstorm series where it can help you for all Pharma competitive examination. Only one series for all Pharma competitive examination. Yes, in this video, I'm going to discuss about the classification of crude drug from a subject called pharmacognosy, right? And also at the end of this session, we're going to have a multiple questions regarding this topic, okay, so that you will understand the importance of this subject in all examinations point of view, okay? Yes, but do not forget to subscribe this channel because you are going to miss all the important content in future because we have planned so many series, okay, so many modules in this YouTube channel for all pharma aspirants all over the India. Okay, yes. So classification of crude drug. So we know that the pharmacognosy, the subject pharmacognosy is nothing but study of crude drugs from the natural origin. Okay, pharmacon means drug. Gignosio means to acquire the knowledge. To me, gignosio means to acquire the knowledge. Yes, exactly. So what do you mean by pharmacognosy? Firstly, pharmacon means drug, and whereas gignosio means to acquire the knowledge of crude drug, which means in this entire pharmacognosy subject we are going to discuss about, we are going to learn about the crude drugs, right? So the crude drugs may be of they are obtaining from several sources: plant sources, animal sources, marine, mineral, micro plant, tissue culture, right? And there are several sources, but they are huge in number. They are huge in number. We can't find, and we cannot find an exact number, but they are very huge in number. Okay, more number of sources are there, more number of drugs are there. But is it possible to remember and distinguish easily everything? Is it possible to remember it? Is it possible to remember each and every drug if they are not having any classification? If they are not having any classification, it is difficult to remember each and every drug, and it is difficult to handle. Is it clear? And that's the reason, because they are huge in number. The plant drugs are huge in number, the animal drugs are huge in number, the minerals, everything is huge in their number, right? And they're wide sources they are. That's the reason, to remember and for ease of handling, right? We are classifying them into different types. Okay, so the classification of crude drug can be done by this many types, sir, but it is somewhat difficult to remember. But what the speciality of your GDC is? Don't worry, I'll tell you one simple code because the GDC is the most familiar for the smart work or smart study. Do remember this code, right? ATM PCCS, right? We do have a ATM cards. For that, we do have a pin code. Okay, that need to be changed successfully whenever we require. ATM pin code change successfully, or ATMPCCS, whereas A stand for alphabetical, T stand for taxonomical, M stand for morphological, P stand for pharmacological, C stand for chemical, and C stand for chemotaxonomical, and S stand for serotaxonomical. Yes, this many times you have classified the crude drugs because they're huge in number. But each classification has their own importance. Because alphabetical, yes, our drugs are. The name itself will tell you about the classification. A stands for alphabetical, which means we have classified the drugs alphabetically, based like simply like a dictionary. Okay, and yes, taxonomical based on their order, superorder, like how, and pharmacologically based on the pharmacological action that they are producing. Okay, and chemical based on the chemicals that they were containing. Okay, morphological based on the morphology, whether it is a leaf or root or fruit or bark or stem. Okay, and lastly, chemotaxonomy. There is a combination of both chemo and taxonomy. Okay, the combination of both, which means in specific family plants, they do contain a specific drugs, that is a chemotaxonomical feature. Okay, yes, let us see each one in detail. Okay, let us see each one in detail. You can have an example here itself, chemotaxonomical, because it's a special one. Okay, in specific families, they do contain a specific secondary metabolite. That is a fixed one. That is a fixed phenomenon, which means all the Solanaceae family plants contain tropanonucleates, whereas Solanaceae is a taxonomical feature, and tropanonucleates is a chemical feature. Most of the Umbelliferae families contain volatiles. Clear? So the Umbelliferae is a taxonomical feature because family is a one of the taxonomical feature, taxonomical order, superorder, family, genus, species, because that is a one of the taxonomical feature. Okay, chemo means chemicals, right? Like how, based on, see, what I want to say is, chemotaxonomy means if you want to get a drug, if you want to prepare a medicine, or if you want to prepare a dosage form based on the chemical constants, if you directly go for this classification, okay, you can find out, okay, what are the drugs, what are the families containing this type of secondary metabolite. Okay, if you want volatile, I can simply go with the I can simply go with umbellifera family, right? And if you want propanol cuts, I can simply go with Solanaceae family. Like how, okay, chemotaxonomical classification or biochemical classification will help us. Okay, let us see. In generally, what do you mean by crude drug? Before going to understand about classification of crude drug, you should know what is crude drug. These are the products from plant and animal and mineral origin found in raw form, okay, not a processed form, except collection and storage. Okay, if you have collected any plant part, right, maybe I'll tell you as an example that, okay, what is a example of a crude drug? If you have taken, okay, if you consider the rhizome of ginger, rhizome of turmeric, okay, if you consider the rhizome of turmeric, that could be the that could be uh, crude drug. But whereas if you convert the rhizome into powder, that is turmeric powder, but it's a processed one. Okay, it is a processed one, that is a drug. Clear? Is it clear? Yes. So which is a raw form? Any plant part or animal part or mineral part or animal or marine or mineral. Okay, any plant, animal, mineral or marine which is in a raw form, except collection and drying. If it should not go any of the process, except collection and drying, it should not go any of the process. Clear? So such type of plant parts, animal parts, mineral parts, but marine parts, all these are considered as a crude drug. But one important thing is that, but all these sources should contain, okay, which is the plant part or animal which is considered as a crude drug should contain one main important property, that is medicinal property or pharmacological property. Okay, that's the reason. So if it is not having a medicinal property, what is the use of making into dose form? Is it clear? Yes. So the crude drugs are obtained from the different natural sources in the treatment of wide spectrum of disease. Exactly. They should have a wide spectrum of disease. They should be used in the treatment of wide spectrum of disease. Okay. And therefore, the adequate study is necessary to arrange them in a scientific and systematic manner. Exactly. So we do have a wide spectrum. They are wide spectrum. They're huge in number. Okay, do we have only one type of plant? No, we are having a lakhs of species are there, right? So many orders are there, right? In that, so many families are there, so many genus plants are there, so many species, subspecies also there. For that reason, right? So they are, they're difficult to handle. For that reason, we need to arrange them in a systematic and scientific manner. For that reason, we are applying the classification of crude drug. Now, we have classified them into this many types, alphabetical. Do remember one code, ATM PCC. If you have your ATM card, if you have your ATM card, that pin code should be changed successfully. Which means ATM PCCS, which means alphabetical, taxonomical, morphological, pharmacological, chemical, chemotaxonomical, and serotaxonomical. Here, all of the 7 classifications we do have, they have their own merits and demerits. See, not every classification will satisfy all the criteria. See, alphabetical classification. In that classification, we can only find out the classification based on the alphabets, but we cannot found their pharmacological action. Like how in the taxonomical classification, we can only classify them based on the taxonomy, but we cannot classify them. We cannot get the data based on the alphabet and based on the pharmacological action also. We only have a specific data. When it comes to morphology, we only know about whether it is a leaf or root or stem, but we cannot find them in a systematic manner, which means we cannot find an alphabetical manner. Okay, so like how, let us see each one in detail in a very quick manner so that you understand. Okay, alphabetical, right? You can see that is simple for, let me tell you an example. Dictionaries. In the dictionaries, right? What we observe in the dictionaries, apart from the all other study materials or maybe any other books, right? So dictionary is a book, okay, or it is source where you can divide or where you can know where all the data was arranged in an alphabetical manner, like similar to that of dictionary here with the entire drugs here, but it is similar to the dictionary. But here our dictionary is pharmacopoeias. In the pharmacopoeias, if you have studied, if you have opened the IP, right, Indian pharmacopoeia, all the modules or all the editions, they have arranged in alphabetical order only, started with the N, ends with Z, all the letters and all the drugs, okay, based on the revision. Okay, every year all the drugs they have added. Clear? Yes. The crude drugs are arranged according to the alphabetical order. Yes, but what is the use? It is a very easy and quick to find out the drugs what we require. Simply, if you want to, if you want a distillate, simply, okay, open the open the section D, and thereby you can find to the distillate. All the data will be there. Like similar to the dictionaries, right? But examples are, for alphabetical classification, examples are pharmacopoeias, okay, reference books are the best examples, okay, dictionaries as well. Okay, let us see how we classify the alphabetical classification. You can see, okay, all the drugs which are comes under A, acacia, agar, ashoka, arjuna bark, and all the drugs which are comes under the B, benzoin, belladonna, like how classification will goes on based on their alphabetical order. Okay. Yes, so this is an alphabetical order, and yes, from this alphabetical order, do remember what are the examples for the alphabetical order and how we classify them in a alphabetical classification. Examples are pharmacopoeias and reference books and dictionaries. And whereas, do remember these things. Okay, they'll ask you several questions like uh, mostly match the following questions they'll ask you. Match the following questions. Okay, on one side, on section A, they'll give you the type of classification. On section B, which means in front of that options, they'll give you all the definitions or maybe examples like alphabetical classification, acacia, benzoin, caraway, dill, ergot, fennel. Okay, so like how they'll goes on. So try to remember what is the classification meant to be or what was the example for that. That should be remember. Next classification is taxonomy, which means based on natural relationship or phylogeny among all the plants, which means every plants do have phylum, division, class, order, family, genus, and species. And but this was an exact data. This was an exact data where you can find the roots of the particular plant, which means from which phylum, from which order it has originated. From which order it has origin. But what was the use of knowing this origin or maybe the taxonomy? You will know the exact features of the plant, right? So because do they no we have classified them. Firstly, we have classified the entire living organism to flora and fauna, right? So in this flora, again these plants, right? Again these things, okay, can be subdivided into six and more than six orders, right? Each order has their own specific features. Clear? And they in that order they are going to have a different special features, like how, okay, the classification based on their phyla, order, and everything. Yeah, in this simply it's like a clear data like where it has origin. Yeah, so based on that, also based on the family, we can find out, okay, what type of things they do have. Based on the order, we can find out, okay, what type of features, either morphological features or chemical features they do have. We can predict. Okay. Yes. So this is an example for taxonomical classification. Okay, you can see taxonomy of belladonna. Example is belladonna belongs to the phylum of spermatophyta. Division is angiosperms. Right. So it is angiosperm. So based on that, okay, if you want to find out whether the belladonna, okay, is which type of plant, which type of division, it is an angiosperm. Okay, angiosperms have a specific features. Like how if you want to if you want to prepare a medicine based on the angiosperms division, like how we do have a different orders, right? So if you want a specific division plan to make your doses form, simply you should rely on this taxonomical classification. Okay, outer tubiflora and family Solanaceae and Atropa belladonna. Okay, simple. It's a very clear information regarding the drugs or regarding the next, morphological classification, which means morphology is nothing but external features. Okay. Yes. If you want to if you want to prepare a dosage form by means of the leaf, okay, because you have depended on one type of extraction method, right? In order to in order to bring out the active constants, you rely on, okay, maceration process. Let me tell you an example. If you want to prepare a dosage form based on the maceration technique, right? So for the maceration technique, the leaves are more suitable or thin texture plant parts are more suitable. In such times, you need to rely on leaf. But how can you find out whether it is a leaf or root or stem? Based on the morphological classification only. In alphabetical classification, we have not discussed the plant parts. In taxonomic classification, we have not discussed the plant parts. But whereas the morphological classification is nothing but where we are where the entire drugs can be classified into based on the plant parts. Okay, all the leaves under one section. Okay, all the fruits under one section. Here no alphabetical classification, but simple. Okay, all the leaves are categorized under one section. All the fruits are categorized under one section. All the all the roots, all the rhizomes, all the flowers, okay, all the stems. So like how the classification is goes on. So that means to be our morphological classification. Before that, okay, before that, we have classified them into two basic things prior to that of morphological classification. Basically, we have classified them into two types: organized drugs and unorganized drugs. Okay, organized means cells are organized in nature. Am I right? Cells. Okay, if you see any TS or TS or LS of any plant part, okay, cells are arranged systematically, organized manner. That means, okay, if anything contains cell, anything contains, okay, cellular tissues or cells, if any plant part or animal part, if they contains cell or cellular tissues, that could be the organized drugs, which means defined as histological or cellular tissues. Whatever the plant or whatever the thing, okay, what is the animal or living thing, if they contains cells or tissues, okay, that could be considered as a organized drug. So leaves, fruits, everything they contain, all the plant parts contains, okay, all the plant parts or all the animal parts contain cells, and based on that cells they do have a tissues, which means if it contains cell or tissue, it will be organized drug. Okay, examples are shoot, leaf, fruit. So like how we have classified them into, firstly we have classified them into organized section. Okay, in that organized, all these are comes under this organized, like leaf, root, stems, and everything. All the leaf drugs are under one section. All the roots are under one section. Okay, they complete data. But whereas what do you mean by unorganized drugs? Yes, unorganized drugs are nothing but the secretions. Okay, which does not contain any cellular tissues. Okay, so unorganized drugs are nothing but they do not contain any histological properties or histological features, which means they do not contain any cells. I'll tell you example. Okay, leaf is organized drug. Okay, whereas a extract or maybe latex, dried juices, okay, milky gum exudates, all these are comes under the unorganized drugs, which means the plant secretions or maybe the extracts, okay, or maybe the dried juices, all this, whatever it whatever it may be, if it is not containing, whatever it may be, if it is not containing cells, that can be simply considered as a unorganized drugs. Okay, you can see here resins, latex, acacia gums, resins, okay, all this acacia, okay, benzoin, okay, all these are nothing but they comes under the unorganized drugs. Okay, so like how next one, chemical classification. What do you mean by chemical classification? Simple. Okay, based on the, see, if you want to prepare an CNS stimulant, what you require? You require ultimately an alkaloid, right? Or if you want to prepare a CNS stimulant, you require alkaloid especially. Okay, but in order to prepare, see, so in order to obtain a specific drug based on their chemical feature, okay, see, because we have classified the entire secondary metabolites into different types: alkaloids, glycosides, resins, tannins. But each secondary metabolites having their own specific pharmacological action, right? So based on the pharmacological action, they do exhibit. Okay, based on the pharmacological action, they do exhibit. We are preparing the medicine. We are preparing the dosage form. So here, if you want alkaloid drugs, okay, till now we have not mentioned any class. See, till now we have not mentioned about the secondary metabolites or about the chemical properties in the previous classification. But if you want to if you want to get an alkaloid drug, if you want to get a glycosidic drug, okay, so based on the chemical features, if you are classifying the drugs, that could be chemical classification. Chemical classification based on their chemical constants. All the drugs containing alkaloids are under one section. Okay, next, second section, glycosides. Next, tannins, resins, flavonoids. Like how? Based on their chemical properties, based on the chemical constants. Okay, all the alkaloid drugs, Rauwolfia, ergot, opium, ipecac, and cinchona. Right, all those glycoside drugs, senna, rhubarb, cascara, and aloe. Liquid lipids and volatile oils, peppermint oil, sandalwood oil, lemongrass oil, cinnamon. Okay, tannins, pale catechu, like how it goes on. See, simple. They are all under, they are all different books. Simply go. Yes, and for all the alphabetical classification, we'd have pharmacopoeias. For all the taxonomic classification, we'd have some books, right? So, like how for specific chemical classification, also we have a separate books. They are all separate classification. They are not all same. Okay, they have a separate books. They have separate classification. Okay, if you want to get a drug based on the chemical classification, based on the chemical properties, you should rely on the chemical classification based books. Okay, yes, so this is about chemical classification. If you are classifying, if you are arranging the drugs, if you are arranging the drugs based on the chemical properties, that could be the chemical classification. Okay, yes. Next, pharmacological classification. Yes, what do you mean by pharmacological classification? Yes, if you want to prepare a cardiotonic, okay, if you want to prepare a cardiotonic, or if you want to prepare a GI stimulant, if you want to prepare, okay, anti-muscarinic drug, if you want to prepare, okay, drugs acting on CNS. So simply you should go on pharmacological classification. Okay, simple. We have classified, we have arranged all the drugs. Whatever the drugs we have arranged all the drugs in a simple manner based on the pharmacological action. Okay, all the drugs which are acting on CNS, they are also in separate section. All the drugs which are acting on GIT, they are under separate section. Like how we have classified the drugs, if you are arranging the drugs based on the pharmacological or therapeutic response that they were producing, that could be the pharmacological classification. Clear? So you can see all the drugs acting on CVS, that could be digitalis, squill and spanthes. Drugs acting on GIT, senna, castor oil. Okay, the drugs acting on CNS, opium, hyoscyamus and belladonna, and drugs acting on respiratory system is licorice, 𝙂𝙎𝑃𝑆 and ipsum. And drugs acting on ANS. Like how based on the pharmacological section we have classified. Okay. Yes. And next one. So chemotaxonomic classification, we already discussed that. Okay, chemotaxonomical classification, chemical facts of the plants and taxonomic position of the plant. You can see here. Okay, tropinol are found in Solanaceae plant, which means here taxonomy is nothing but a family. Taxonomic feature means family, whereas chemical facts means. See, let me tell you an example. Best example. All the umbelliferous plants contain 𝙂𝙄𝙎𝙎𝙬. Okay. All the rootaceae plants also contain 𝙂𝙬𝙎𝙬. All the Solanaceae plants contain tropinol. Okay. So which means here there is a phenomenon that specific chemical constants can only found in a specific families. Okay. Specific chemical constants only found in specific families. Tropinol are specific alkaloids that can only found in Solanaceae family. That could be the chemotaxonomy, which means specific type of secondary metabolites only found in specific families. Okay, that could be the chemotaxonomical classification. Here you can see the system classification is based on the taxonomy and chemical similarity. Okay, and or link that exists between the different plant elements. Okay, and also it can establish the relationship between the position of the plant and the chemical facts. It establishes the relation between the plants, which means in which plant family, in which family they do contain which type of secondary metabolites. Clear? So that clarity can be obtained by the chemotaxonomic classification. Okay, example are tropinol and Solanaceae family. 𝙂𝙬𝙎𝙬 from the umbelliferae. Like how there are several examples. Okay, yes, this is about the chemotaxonomic classification. Okay, and next is serotaxonomic classification, which means see as like the plants do contain. See what do you mean by chemotaxonomy? Taxonomy means family, chemo means chemical facts, which means in specific families they do contain specific chemical constants. Like serotaxonomy, serology is nothing but study of protein. Okay, serology is nothing but a study of protein. You can see study of protein, which means taxonomy means serology means protein, taxonomy means family. Simple. Correlate these two things. In specific plant species, they do contain specific proteins. Okay, based on the specific proteins in their taxonomy, based on the specific proteins in their DNA, we can separate it. Okay, because so that particular DNA can be used. Okay, based on that particular specific gene. Okay, we are going to prepare some medicines as well. Clear? So that's the reason the application of serology in solving the taxonomic problem is called as serotaxonomy. Okay, so which means study of antigen and antibody reactions is called as serology. Okay, and based on what type of an antigen antibody and what type of proteins are there. Okay, what type of protein they containing. If you are if you are finding out these things, that could be the serotaxonomy. That could be the serotaxonomy. Clear? So here itself, all the organisms which closely related to have the similar proteins, all the organisms which have a closely related would have a similar proteins. Clear? Which means if they are closely related, which means if two families, okay, maybe Apocynaceae or Acanthaceae, if these two are having same properties, which means morphologically if they are similar, okay, if they have the same type of flowers, same color of flowers, which means okay, if they are closely related, definitely they would have a same proteins. Okay, that is the same phenomenon. See, simple. If you are classifying the drugs, if you're classifying the drugs based on the protein that they were containing, simply that could be serotaxonomy. Is that clear? That's it. Simple as like that. Okay. And yes, and it determines the degree of similarity between species, genus, and family. Yes, and also to find out that okay, if they're containing same protein, definitely we can assess what type of degree they do have. Okay, what type of degree of similarity they do have in the class in the particular plant species. So that can be that we can find out. Okay, so this is about serotaxonomy. Simple. So finding out the what type of protein that they were containing, check the similarity between the species and the family and everything. Okay, yes. So that is serotaxonomy. Okay, let us see the most related process of serotaxonomy. Okay, this is how we going to done the we can have the protein extract of the plant origin. Okay, and the antigen is extracted, and also that antigen could be introduced into the experimental animal. Okay, and thereby it produces a specific antibody. Okay, so here we have introduced an antigen, we have collected the antigen from the plant, okay, and that could be exposed to an animal and thereby produce an antibody, okay, and that anti-serum is made. Anti-serum is sorry, sorry, first the protein is made, protein is extracted, okay, and that protein, that means antigen is going to injected into the animal, okay, thereby it produce an antibody. Okay, simple. Plant extract is taken, exposed to an animal, experimental animal, there it produces an antibody. Okay, that antibody, okay, the antibody containing antigen solution, which means the blood serum with antibodies is called as anti-serum. Okay, so anti-serum solution is there, and that anti-serum is made up to react in proteins of the other taxonomy, which is which finds are to be determined. Which means simple. Plant extract is taken. See, whatever the plant extract is taken, from that we have taken an antigen. Antigen is taken. Okay, that is injected to the animal, whatever the animal, okay, for experimental animal, and thereby this animal produce an antibody. This antibody containing solution is called as anti-serum. Okay, simple. Okay, and that anti-serum solution can be introduced into the plant where we want to find out the relativity. Okay, if you're having doubt on this plant, okay, this plant may have the relativity. If this plant is having a relativity to this plant, simply you can inject the same anti-serum blood anti-serum solution, and thereby if it is produce antibodies, simply that could be okay, if it forms antigen antibody reactions, that could be the same plant. Okay, so which means they have the relativity, same protein. Clear? Yes. So like how we're going to done the serotaxonomy. So let us see a few important questions related to this topic. Okay, and thereby we'll end this session. And with the following classification of herbal drugs gives better understanding of biosynthetic pathway of the drugs. What option A? Botanical, chemotaxonomical, taxonomical, and chemical. Okay. Option B is correct. Okay, biosynthetic. What do you mean by biosynthetic here? Biosynthetic means, which means in all the biosynthetic pathways we are going to obtain the secondary metabolites. Okay, we are going. So which means in specific plants they do contain specific secondary metabolites. Which means this is a relative phenomenon for the chemotaxonomical feature. Biosynthetic pathways in the drug. Okay, clear? Which means if you are getting simple example, I'll let you know. I know you're confused. So if they are containing morphine, okay, they should get, if a plant is containing opium containing morphine, for the morphine, okay, in order to synthesize the morphine, they do undergo for the 𝘂𝙾ic acid pathway. Clear? Which means if they're having a specific secondary metabolites, they should have a specific pathways as well. Are you getting my point? If they are getting a specific secondary metabolites, they are having a specific pathway, 𝘂𝙾ic acid pathway, acid malonate pathway. Like okay, for better understanding in which of the classification having the clarity regarding the biosynthetic pathways and the chemotaxonomic features only, in the chemotaxonomic classification only we can understand, because we are saying that in the chemotaxonomic classification, okay, this plant species containing this particular secondary metabolite, which means if that particular secondary metabolite is present now, which means they are having specific pathways. Okay, propane found in Solanaceae member family are similarity in, okay, obviously we have discussed propane. Okay, alkaloid found in Solanaceae member family are similarity in morphology, chemical, chemotaxonomical, and serotaxonomy. Chemotaxonomy, obviously we have discussed, right? Chemotaxonomy, okay, I tell you, okay, taxon means family, chemo means chemicals or chemical constants. Choose the incorrect option based on the chemical classification. Okay, so Based on the examples also we are going to get the question. Okay, what type of drugs they contain alkaloid, what type of drugs, what are the example for distal, what are the example for glycosides, all these things we need to consider. Okay, alkaloid drugs, datura, tannin guargum, resin turmeric, and glycoside chirata. What is the wrong option? They're asking, right? Read the question carefully. What is the incorrect option? Okay, undoubtedly tannin. Okay, guargum. Okay, exactly. Which type of classification is based on the protein the plant contains? Just now we have discussed. Obviously, undoubtedly, B, serotaxonomy, because alphabet classification won't tell you about protein, morphological classification won't tell you about protein, chemotaxonomy won't tell you about the protein. Clear? That's the reason. Serotaxonomy is the right answer. Okay, yes. Last question of this series: which of the following is not an organized drug? Not organized drug. Not organized drug. Fennel is a fruit, colophony, okay, 𝙂𝙀𝙎𝙎𝙬𝙀 and senna. What is the right answer here? Which is a not organized one. They're asking that which is an unorganized one. Okay, read the question carefully. Not an organized one, which means they're asking which of the following is an unorganized drug. What is the right answer? Undoubtedly, okay, colophony. Colophony is the right answer. Clear? Yes. So these are the few example of these questions from this topic, classification of crude drugs. Okay, I hope you like it and I hope you understand. Okay, for more such content, for more such type of videos, do subscribe this channel and do follow this Pharma Thunderstorm series. Okay, so we have prepared these questions and we have prepared the content based on the all examination point of view. Because GDC is not only meant for GPAT, GDC meant for the pharma competitive examinations. Okay, all pharma competitive examinations. Clear? Yes. Thank you, aspirants. We'll see you in the next class.